See, let us have concept about repetitive DNA, that is the part of genome which gets repeated so many times, the type of repetitive DNA, how they are organized in the eukaryotic genome, what kind of function they perform. All these salient points we can learn in this presentation. So eukaryotic chromosomes show complex organization characterized by repetitive DNA. We find that in case of eukaryotes, there could be a specific number of chromosomes, and such chromosomes are made up of individual DNA molecules. And if we observe the DNA itself, we find that there are defined areas in DNA. Certain portions encode for a specific protein. They are coding regions. They are protein coding regions. And then there are regions which are non-coding areas. There could be repetitive DNA sequences. So, but in case of prokaryotes, the genome contains main bulk of DNA, which is devoid of satellite DNA. It means very much difference exists between eukaryotic and prokaryotic genomic organization. In case of prokaryotes, the genome is quite simple. Major portion is defined to encode specific proteins, because everything will have to be accommodated in short portions of the genome. We can classify the repetitive DNA into different types. For example, there could be two major divisions of repetitive DNA. One is highly repetitive, that portion which has maximum repetition of nucleotides. Such portions are represented in the form of satellite DNA. And then there are middle repetitive portions, moderately repetitive portions, which could be classified as tandem repeats and interspersed retrotransposons. Now, tandem repeats are further grouped as multiple copy genes, like ribosomal RNA may be represented so many times in the eukaryotic genome. So such portions are multiple copy genes. And then there are mini satellites and microsatellites. The mini satellites, which are also referred as variable number tandem repeats, that is VNTR regions, they are scattered all along the genome. Exactly the same way there are microsatellites, which are also called as SSR, that is simple sequence repeats or short tandem repeats. And then there are interspersed retrotransposons, which are actually transposable portions of the DNA. They get moved from one portion to the other in the chromosome. Such segments in DNA are called as signs and lines, that is short interspersed elements or long interspersed elements. Signs are represented in the form of LU family in case of humans, and in humans the lines are represented in the form of L1 family. So we shall understand about all these different types one by one by going in detail about these different types of repetitive DNA. Just for understanding, I have drawn here two different chromosomes, and suppose these two chromosomes are represented in the form of two DNA molecules, means these are two separate DNA molecules. In one chromosome, the different repetitive portions are shown in colored form, like the centromeric areas are there. Here is the centromere, suppose, and this centromere, suppose this portion is the centromere of the chromosome, or this is the portion in DNA which is demarcated for centromere. So here the satellite DNA will be present. In the same way, at the terminal ends, that is telomeric portions, also contain satellite DNA. So major part of satellite DNA will be confined in telomeric portions, that is on the ends of chromosome, as well as in the centromere. So every chromosome will have its telomere and centromere because these portions are well distinct, and that is the portion where satellite DNA will be mainly present. Besides that, there will be micro and mini satellites areas and signs and lines. So all these are scattered throughout the length of the DNA molecule, and almost every chromosome or every individual DNA molecule will have these portions spread along their length. So this diagram actually shows distribution of repetitive DNA in the genome of eukaryotes. Here certain points you can remember, like highly repetitive and moderately repeated DNA constitutes up to 40% of human genome, and a very small part of genome actually codes for protein in majority of eukaryotes. So quite good amount gets represented by repetitive DNA. In case of sea urchin, 20,000 to 30,000 gene encoding proteins occupy less than 10% of the genome. So we can see that how much representation for protein coding gene is there in the entire genome in case of sea urchin. In case of Drosophila, only 5 to 10% of genome is occupied by genes coding for proteins. And in case of humans, 20,000 to 25,000 functional genes occupy less than 5% of the genome. See, let us have idea about satellite portion of the DNA. The satellite actually refers to the genomic DNA separated in a test tube as a prominent layer of bulk DNA from accompanying satellite layer of repetitive DNA. So we can see there would be two different portions of DNA. If we separate DNA, we find that the major bulk is that portion, and this major portion is actually DNA, that is bulk DNA, and a small portion will be quite separated from it, and that is referred as satellite part. So the satellite part, as I just said, that will include mainly the DNA portions confined in the centromeric and telomeric portions of the DNA. Then in this diagram you see this, suppose these are two separate two different DNA molecules. Okay, and if you see on the either ends, here is telomere, this side it is telomere, and this is centromere. So this much portion will be mainly heterochromatic. It will be formed by constitutive heterochromatic portions, and they have repeated DNA sequences. So satellite DNA, that is mainly represented in the form of telomeric portions. Centromeric areas are also contain satellite DNA, and these portions are mainly heterochromatic. They have condensed arrangement of nucleotides, or you can say the genome is much condensed in such portions. So heterochromatic portions. See, I'm going to explain about the characteristics of satellite DNA. Heterochromatic portions like centromeric and telomeric parts contain segments of DNA repeated many times. For example, in case of Drosophila, 10 base pair sequence, and these 10 base pair sequence is AAT, then AAC, ATAG, is repeated randomly many times in centromeres of all 4 chromosomes. Drosophila possesses 4 pairs of chromosomes. So if we consider 4 individual chromosomes, then they will have in their centromeric portion this 10 base pair sequence repeated so many times. And regarding telomeric region, see, telomeric hexanucleotide sequence, that is GGGGTT, are represented more than 50 times in tetrahymena, which is a unicellular eukaryotic ciliate. So here in the telomeric portion of this animal, you find the presence of repeated DNA sequences. Then in case of humans, the sequence GGG ATT is repeated many times in the telomeric portion. So as I said, satellite DNA that will mainly be represented in the form of constitutive heterochromatin, and that will be mainly confined in the telomeric and centromeric areas with so many nucleotides repeated several times. Let us have idea about multiple copy genes, because when we consider moderately repeated portion in the genome, then we come to know about multiple copy genes. Some functional genes are repeatedly duplicated, like ribosomal RNA genes. See, the ribosomal RNA genes are located in nucleolar organizer region of the chromosome. Nucleolar organizer region means it is secondary constriction, and that portion is actually it is adhered with the nucleolus present in the nucleus. So such ribosomal RNA genes are located in nucleolar organizer regions of the chromosome and are duplicated several times so that they can provide excessive RNA molecule for the formation of ribosomes, because ribosomes are such a structure in the cellular system which are made up of ribosomal RNA and a number of different types of proteins, and they help in protein synthesis. The other point you should know that in case of Drosophila, 120 copies of ribosomal RNA are present per haploid genome, and in case of humans, multiple copies of this gene are clustered on the short arm, that is P arm, of chromosomes 13, 14, 15, 21 and 22. These are actually acrocentric chromosomes in case of humans, and they all have secondary constrictions, that is nucleolar organizer regions, and from such regions the ribosomal RNA is actually synthesized. You should know that this single gene unit encodes for a large precursor molecule, that is 45S ribosomal RNA is synthesized from that portion of ribosomal RNA gene, and that further gets processed into the types of ribosomal RNA like 5.8S, 18S and 28S. We know that another 5S ribosomal RNA is also required, but that is made available from a different portion of the chromosome. Now the other type of moderately repeated DNA sequences are mini satellites and microsatellites. Let us have idea about these two. The mini satellites are also highly repeated DNA segments. They are nearly 10 to 100 base pair repeats, typically repeated 5 to 50 or even more times. Generally, they are rich in GC bases, and they are also known as VNTR, as I just said, that is variable number tandem repeats. And in human genome, mini satellites occur at more than 1000 locations. They show high mutation rate and high diversity in the population. So in DNA fingerprinting, we mainly use mini satellite portions for identification purpose, or just to find out the culprit present at a particular crime place. The microsatellites are also of use. They are used to analyze individual characteristics. They are used to analyze the characteristic of populations. Microsatellite segments are also highly repeated, and they possess 1 to 9 base pair repeats, mostly repeated 5 to 50 times or even more. And microsatellites account for approximately 3% of human genome, generally rich in 80 bases. They are also referred as SSR, that is simple sequence repeat, or short tandem repeats. So in the genome you see, suppose these are mini satellite portions which will have 10 to 100 bases repeated 5 times or 10 times or 50 times or even 60 times, and they are scattered all along the genome. In case of microsatellites, the repeats will contain 1 to 9 base pairs, and such bases will be repeated 5 to 50 or even more in the genome. So through this line diagram, we can have idea that such segments in the DNA are also present in quite good amount. Finally, we should know the types of repeated DNA like signs and lines, that is short interspersed elements and long interspersed elements. These are found almost all eukaryotes. In all eukaryotes, signs and lines are found. They contribute in the evolution of the genome. We have found that such areas are transposable in nature. They can move from one place to the other and can make certain genes active or inactive. Means such kind of characteristics they have. They are transposable genetic elements. In case of humans, they account at least 34% of the genome. And signs and lines DNA segments invade new genomic sites using RNA intermediate. It means suppose there is a part of signs, it is capable to get transcribed, and its RNA will then change into DNA, and that DNA will get incorporated in some other place of the genome. Exactly the same way, lines are those segments of DNA which are actually longer, and they can also get incorporated to so many other places in the genome. See in this diagram, suppose the upper portion contains signs. So you can observe that these portions in the genome, they would be 100 to 300 base pairs, and 15% of the genome in humans gets occupied by signs, that is short interspersed elements. In case of lines, these portions in the genome may contain 5,000 base pairs, and they are actually autonomous segments as they code for proteins, and they are usable for their transposition. It means this much portion, if it is transcribed into RNA, then that RNA can undergo translation process to form the reverse transcriptase enzyme. It can also synthesize the endonucleases or integrating enzymes. So such portion will be capable to synthesize its own usable contents, and then it can change into DNA, and that DNA segments will get incorporated in some other portions of the genome. So when they move from one place to the other, then and they can move close to a gene and can cause its inactivation, or sometimes may activate that gene to start its function. So and if they get inserted at several places in the genome, then definitely they will affect the size of the genome. So they play important role in the organization of eukaryotic genome as well as in the evolutionary process. Now, if we consider the important characteristics of signs, we find that such portions are less than 500 base pairs and base pairs long, and may be present 500,000 times or more in human genome. LU family, LU1 is a restriction enzyme. So mainly the portions, that is 500 base pair portions, contain area where this LU1 restriction enzyme can cut, means it's a recognition area that is found to be present in the signed portion. So LU family of signs are well characterized, 200 to 300 base pairs long, and are dispersed uniformly throughout genome, both between and within genes. And this is all being explained regarding human beings. LU family encompasses more than 5% of entire genome in case of humans. Regarding lines, that is long interspersed elements, the lines are another category of repetitive transposable DNA sequences, as I just mentioned. And there is one specific kind of sequence which is referred as L1 family, found to be scattered throughout the genome of humans. So L1 family in humans even comprises about 64,000 base pair long DNA present up to 100,000 times. The line's DNA segments invade new genomic sites using RNA intermediates, because when it has to go to another region, then first it will be transcribed. So RNA intermediate will be used so that RNA could change into DNA in the presence of reverse transcriptase, and then by using integrating enzyme it gets inserted to a different location in the genome. The L1 DNA sequence is first transcribed into RNA, which serves to synthesize DNA using reverse transcriptase. And reverse transcriptase is also encoded by L1. It also encodes integrating enzymes. And the enzyme available from lines can be used for the transposition of sign segments. So these are some of the important points regarding repetitive DNA, and I have explained all such things in very concise way. One can have better idea by going through some textbook knowledge.